Hi, my name's Alex. I'm just making this video because I talked to Professor Ice, and he had mentioned that people were wondering what to do with Sangamo Therapeutics, a stock I had selected for the URI RAM fund. And I just sort of wanted to get my thoughts out there and sort of explain why Sangamo Therapeutics is a good investment. Right off the bat, has a $1.63 billion market cap right now. It's a gene editing company, and they have a really cool technology called zinc finger nuclease that allows them to, and they have a patent on it, and basically it is the best gene editing solution out there. Like, there's a lot of techniques, and you've probably heard of CRISPR. There's a few other ones like TALENs and stuff, but ZFNs, zinc finger nuclease, are beyond good. So I'm going to play a quick video that should explain.
Zinc finger nucleases are restriction enzymes made of a chain of zinc finger proteins with a FOC1 endonuclease attached at the end of the chain. They are used specifically to select a sequence of nucleobases in a DNA strand and induce a double-stranded break at that site. Zinc finger nucleases must dimerize around the strand.
So pretty much you have these two things called zinc fingers, each one's a finger, and they bind to a certain part of the DNA that's specified. You can design them to be very, very specific, and the two ends of them sort of come together like scissors essentially and do what's called the double-stranded break, where you basically just cut the DNA. And you can also have the correct DNA also injected with the ZFNs, and the body will naturally take that corrected gene, and when it's rebuilding its DNA, it will have the proper DNA fix in there. So that's the idea of zinc finger nuclease. And what's that useful for? So there's actually a bunch of diseases that are called monogenetic diseases, which are very, very easy to edit. Like zinc fingers can actually do multiple edits, but the easiest ones to go after are diseases that are just like one gene that's messed up. So if you look here, here's just like a quick list of some of them. This isn't all of them, but it's pretty bad, and they're all pretty deadly. So for example, in Canada, it's been estimated that monogenetic diseases may account for up to 40% of the work of hospital-based children's practice. So it's a pretty rough area of diseases, and the impacts are pretty awful. So here are the diseases that Sangamo is officially working on right now. So you have hemophilia, MPS 1 and 2, sickle cell, Huntington's, a bunch of them. So this is actually a lot of products that they're working on compared to a lot of other companies with a similar pretty cheap valuation of $1.5 billion. And I actually made a presentation about gene editing for the RAM fund that I think ICE had shown the healthcare group, or at least mentioned it or something along those lines. And these are my slides for it. This is the zinc finger slide. So it's the same idea where you have two fingers come together and cut, and the efficiency of editing is 99.5, which is insanely high, and off-target cleavage is essentially if the editing makes a mistake. So if the genes are cut in an area that you're not targeting, that's considered an off-target. And recently Sangamo has just improved their zinc fingers to essentially remove all off targets. Like, they say over 1,000-fold reduction, but it's basically because they couldn't detect any. They don't want to say there's none because obviously, there could be some that they somehow missed, but they pretty much looked everywhere and couldn't find any. And it's been tested in animal studies, both primates and mice. Now, the good thing about DNA testing with animals is DNA sort of is the same. Obviously, the contents are different, but the structure and how it behaves is pretty consistent across animals. So here is data from mice that were treated with zinc finger nuclease. Essentially, the mice weren't making a thing called fix. See on the y-axis here, FIX. And so the top left one, the green line shows the measured levels of FIX in the blood. That was the control, and the blue shows the levels of the mice that were treated with ZFNs. So obviously there's a big difference. You went from 0% to 50% of normal, so clearly it worked. You look on the top right, and another sign that it works is as you increase the dose, the amount of FIX, the thing that editing is supposed to fix, goes up. So obviously, if you increase the dose and it went down or stayed flat, that might not be a good sign, but as you take more of something, you sort of want the effect to be more, and that's a good sign. Also, you look at here, baseline was below 25, and week 2 of the edited was 1,000, and that's sort of the same idea here. So clearly the editing works, and it's able to produce the thing that's missing. In this case, it was an FIX thing. So yeah, that's sort of the idea. And the beauty of the zinc finger nuclease is it gives Sangamo this platform called IRVP, or in vivo protein replacement therapy platform. In vivo just means like it works inside of you, like ex vivo, or is like when you take it out of the body and do stuff to it. So it's essentially saying they're able to make proteins that so DNA encodes for proteins. So whenever you have a DNA malfunction, your body reads the DNA as like an instruction manual to make proteins, and when there's a mess up, it doesn't make the protein correctly or at all. So you're essentially able to edit the genes to make the protein that should be there that's not there. So for example, with hemophilia, your body, if you have hemophilia, your body doesn't make a protein that's responsible for blood clotting. So if you're a hemophiliac and you get cut, you sort of just keep bleeding out, and it's pretty bad. You have to take a bunch of meds and get injections all the time and stuff. And another disease that they're targeting is called MPS. It's essentially your body naturally creates these things called GAGs that are just carbohydrates or sugars, and everyone who's healthy has an enzyme that's produced by genes, obviously, that is able to break down GAGs. But if you have MPS2, your body doesn't make that protein or enzyme, so the sugars or carbohydrates or GAGs, whatever you want to call them, just keep building up. And because your body has no way to get rid of them, the cells end up being like real big and overloaded with it, and it causes a bunch of big issues. And you have to get injected with the enzyme that breaks it down every week, but it still doesn't really work well, and you pretty much have an awful life and die, which is not great. So the interesting thing about the protein replacement platform is that they edit a certain gene called albin. And basically the good thing about that is it's highly expressive, which means that gene area is makes a lot of it. So some genes, for example, aren't read very often, and they're not really used that much. Whereas the gene sequence that they target and replace with like a working copy of something, it's always being used. So you only have to edit a small amount of the cells, and then the cells sort of just pump it out. So highly expressive just means when you make the edit, the protein or enzyme that you want to make is just being pumped out of the cell, which is good. And they're found in the liver, so that's what this means. And it can be worked safely because it's a phenomenal target for therapeutic gene expression. So that gene, albumin, don't really know how to pronounce it, but I think it's albumin. That is altered permanently to produce corrective levels of a protein that addresses enzymatic shortcomings. So if hemophilia, you're not getting the clotting thing. MPS2, you're not getting that sugar breakdown enzyme. You alter it, and it pumps out those into your blood. And I can pull it up here. So this is a little document. A lot of this stuff I found through different research groups and stuff. Some of it's my own, some of it is research I've taken from other people, but it's all sort of relevant. This video is about 10 minutes long right now. I'm going to try to make it under like 20 or 30, but you can, it's pretty useful. So like, 30 minutes of your time for a big thing like this isn't too much work. And if I want to make it quicker, I think I would make it more confusing, so whatever. Basically, a very small fraction of the albumin genes replace a missing protein at normal levels, and it's very effective. So it produces a huge amount of protein. Those with one defective albumin gene who only produce 50% are fully normal. So because you can edit 1% of them, then you're not really causing any issues. So like if albumin was very sensitive, like if you didn't have enough, it was really negative for you, that would be bad to edit it and change it with something else, but it's not really needed that much, so you can sort of edit it and have it pump out whatever else you want. That's sort of what that's saying. It's going to be used for disorders where the gene product is produced on a continual basis. It's going to be produced from the liver. This platform should result in completely normal function in those conditions for which it's suitable. So it literally will cure you. There's a few benefits. There's a massive reduction in costs. Pretty much you already know the DNA sequence you need to target. It's just by computer algorithms. And it's as simple as just, slightly altering the target, but it's the same thing. They're all zinc finger nuclease. So normally with drug development, you have to spend billions and billions of dollars developing these, really complex pills that, chemicals you take, and it's really expensive and it might not work. Whereas zinc fingers, all you have to do is just like change a few things around and it can target a whole new disease and can cure a whole new disease for literally like 1% of the costs. There's also a huge reduction in cost of running clinical trials, so that's also expensive. But essentially, the trial is deemed a success if there's protein detected in the blood. So it's pretty easy. You just have to give the patient the dose, wait a little while, take their blood sample or take a few measurements and see if it works. Also, because the diseases that targets are pretty awful, the FDA is pretty eager to approve things quickly. So like, if hundreds of children a year are dying of a disease, the FDA is not going to play hardball and, take a real long time to approve it. If there's a cure for awful disease out there, they sort of, there's things like fast track, priority review, stuff like that. Once essentially you have a few successes, so like, let's say the MPS 2 trial is a success, they have like one more success. At that point, the FDA would be pretty confident that, there's no safety issues, it seems to work. So the approval process will be even faster for more diseases. It's like, once it's proven to cure 5 diseases, perfectly with no side effects, the FDA is not going to, require the same proof for the 6th disease, and patients are going to be real eager to get it, so that'll speed things up. The odds of success from a point of selection of the target are very close. So it's essentially, if the gene editing works, then there's literally no reason why it won't cure the person. When you do drug development and you have a pill and it has to be digested and, there's complex chemical reactions going on and you're not really sure if it'll work. But, if you can literally just edit the DNA and then your body makes the medicine, and it's 100% guaranteed to be right since it's just, reading DNA, which is how the body does things, then as long as the editing works, the cure should work. I showed you some of the animal model stuff. So this is sort of a similar thing to what I said. Results in animals are far more likely to be accurate representation of results in humans than animal results usually are. That is because there's significant differences in like metabolisms and other aspects of the body between animals, but genes are pretty much genes across the board. And since genetic changes result in the exact replacement of the missing molecule that is the root cause of the conditions, it is almost inevitable that if a platform accomplishes the genetic change, the therapy will be effective. Let's see here. So it's not just monogenic diseases. Like I said, there's also other diseases that maybe there's a couple different genes that are causing the mess up. One you can sort of help by just reducing one of the reasons, or now Sangamo has multiple edit zinc fingers where you can fix multiple areas of the gene sequence to cure more things. But the monogenetic ones are plenty to value Sangamo at insanely high. Like if you're able to cure all monogenetic diseases, you're looking at like a $50 to $100 billion company just in those areas, never mind the cancer possibilities and stuff I'll get into later. So yeah, that's sort of the benefits of the zinc finger in vivo protein replacement platform. And again, here is a link to the animal study. I'm not going to go into this, but metabolic pathways most drugs are involved in are far different between the species. For example, pathways involved in food metabolism are often significantly different because the differences in diet between species. Those differences are the reason of the disparity of results between animal trials and humans. So most drugs have an animal trial aspect to them, and people, they're somewhat useful, but a lot of times things behave in animals differently than humans. But since the genetic mechanisms between species differs far less, the results of drugs that operate through them are far more likely to have consistent results in animal and human trials. And that, comes from a mice study. Quickly, I guess, go through this. We investigate whether zinc finger nucleus transgene insertion into the albumin locus could represent a general strategy to express a wide variety of transgenes in the livers of mice. They received the drug and they exhibited stable levels of fixed levels, 60% of normal. We treated mice with the drug, and we detected protein expressions in all four enzymes by Western blot in the liver. So essentially they put in four different types of genes, one that will produce galactose or galactodice, biology things. So this one, which is MPS2's enzyme, and this one, and all four of them were detected in the mice when they weren't beforehand. So it just goes to show that once the platform works, it's as simple as just changing the instructions a bit to be able to produce things that cure multiple different kinds of diseases. So, for example, here's a few diseases that are targets and fully owned by Sangamo. This study could be enough to attract partners for these, given that if the platform produces these therapeutic proteins in humans as in mice, the therapies must be effective. And going off that, there's already some partners. Huntington's disease has Shire as a partner, Bioverative with these two, and Pfizer with hemophilia A, and so, partners are already getting pretty excited about things. So that is Sangamo as an overview, just under 20 minutes in. And recently, if you have been looking at the stock, which you probably have since Ramfund was maybe started when this happened, they had an announcement on 9/5 of data. The stock dropped 31.5%, which was ridiculous. And since then, it's climbed back up 23%, so it's about to where it was before the big run-up, before the news event. Now, I'm invested heavily in Sangamo, and when I first woke up and saw the news I just read the results and read the trial readout, because that's what it was. It was a clinical trial update of MPS2. I was super excited. I was like, oh, I'm rich. The gene editing worked, blah, blah, blah. And then I saw the price action pre-market and was like, oh, never mind. Lost half my money since I had leverage with options as well, like 3/4 of my money. But I'm not too worried. I'll make it back in December and January. In February when they announce the further results. But anyway, nonetheless, here's the press release. So let's see here. they announced the 16-week clinical trial results, including reductions in GAGs, which are these things. That's the sugars I was talking about with MPS II, where there's a buildup in sugars. And it's a phase 1/2 trial evaluating a zinc finger gene editing for MPS II. So they only talked about the low dose and mid dose. In February, they're going to release the data for the high dose. The high dose, by the way, is 5 times the dose level of mid dose. So the mid dose is twice the low dose, and the high dose is 5 times the mid dose. So the difference between low dose and high dose is 10 times. Between medium dose and high dose is 5 times. So there's basically 2 things they measured. It was urinary GAGs. So when your body is not able to break down these GAGs, a lot of times you can see the levels in the urine, and you can measure the amount you're seeing in the urine to get an idea of how much is in the body. So there's a few different types of GAGs. They gave measures of 3 different ones. So the total GAGs declined by 51%, dermatan sulfate by 32%, and heparin sulfate by 61% in cohort 2. There's also been no negative effects, like there's been no health effects that are negative, like no allergic reactions or anything. Let's see, so at 16 weeks post-dosing, mean reductions were observed at 51%, 32%, and 61% of different levels of GAGs. Now, the issue is that, let's see here, they basically, plasma IDS. This is why there was a sell-off. So if you're curious about the sell-off, this is why. At baseline and for the first 16 weeks post-dosing, plasma IDS activity, which is the enzyme that the edited liver cells are supposed to be pumping out, so measures of that enzyme was below the level of quantification of the current assay. So assay is just like a test. And essentially they said they couldn't detect it because it was below the levels of quantification of the current assay. So they didn't detect any. So people saw that and were like, maybe these GAG reductions are from something else, which I'll get into later why it doesn't make any sense. Why aren't we seeing the enzyme that's supposed to be produced in the blood if the gene editing worked? The gene editing didn't work. Let me, sell my stock or whatever. And it's just really not true once you look into it. Like a lot of people who, maybe didn't spend as much time in Sangamo or have a bunch of other investments just saw, what happened on Twitter about people talking about it in the quick headline that there was no enzyme produced and, sort of forced down. I think people are realizing that now since, if I would have not been 100% in and had some cash on the sidelines, I was trying to buy more and you would have had a 23% run up. As people sort of realized they overreacted, and I think that's going to continue. But let's see here. Where am I? Here we go. So let's get to some interesting stuff. So in MPS II patients, we know that when the enzyme, the IDS enzyme, is infused intravenously, it is rapidly taken up by cells and breaks down toward GAGs. We've previously hypothesized that even very low amounts of IDS secreted continuously into the circulation could be adequate to reduce GAGs and potentially provide the first indication of efficacy. And that's a doctor who's an MPS II specialist, literally the top person about MPS II, and he was administrating some of these trials. So in the study, the observed reduction of across total GAGs and the 2 different kinds is encouraging. We hope to understand the clinical relevance of these changes, where they're gonna. So the patients right now, because it's so bad of a disease, are on weekly enzyme replacement therapy, where every week they're just given a truckload of the corrective enzyme in their bodies, like just an insane amount. They just try to flood them, and they have to do that because the enzyme sort of breaks down quickly in the blood. Like it doesn't last long, only about an hour. So you just sort of try to flood and, have as much of an impact as possible and then send the person on their way. But every week they have to come back since by the end of the week, well, really by the end of the session, all the medicine's gone and it's just sort of a temporary solution. It's like spritzing a fire with a water bottle every week. It doesn't really put it out. It just sort of dampens it. So basically, it's supposed to treat it by inserting a new copy of the IDS gene into a precise location in the DNA of the liver cells to enable a patient's liver to produce a continuous and stable supply of the missing human IDS enzyme. The SB913, which is the name of the drug, is a single IV infusion of basically viruses that have the zinc fingers and the IDS gene in it. The viruses inject the zinc fingers and the working copy of the gene into the cell. So once unlocked inside the cell, the ZFNs identify, bind to, and cut the DNA in a specific location in the albumin gene, and then using the cell's natural DNA repair process, Sangamo believes liver cells can insert the proper working IDS gene at that location, and then driven by the strong albumin promoter, like I said before, it really puts out a lot of stuff. The IDS gene would then express IDS enzyme and would be taken up by tissues and cells where the enzyme breaks down, gags. So, on the outside of cells, they have receptors to find the enzyme and then bring it into the cell to absorb it, and these patient cells are really hungry for that enzyme. They haven't really seen it much since the person doesn't make it. Let's see here. If long-term data for the study continues to be positive, the genome editing has the potential to bring tremendous medical progress for MPS II and other monogenetic diseases. So if we look here at the chart, actually, let me get really quickly the other slides that has a more easy to understand situation here. So this is cohort 1. This is the low dose. As you can see, there's not really any change. Sometimes it went up, sometimes it went down a little bit. Not much though. If you look here, all of them are consistently in both subjects and very low. So if you graph that, there's the low dose, red line's the baseline. So they pretty much just hover and bounce around the baseline. If you look at the high dose or the medium dose, cohort 2, they start off at the baseline and they never ever get back up to it, and they're constantly below it. So this is total gags, this is DS gag, and then this is heparin sulfate. So yeah, that's pretty much that. This sort of shows you an idea of what a normal cell versus an MPS II cell looks like. There's all these extra sugars in there that aren't being broken down. And essentially you edit the liver cells, they spew out the enzyme, it travels through the blood, it gets absorbed by cells, it breaks down the sugars, and the person doesn't live an awful life. So these are the results, and I think gag reductions are enough to say that it worked, and I'll bring up some information to sort of show why. So analysts were worried about the IDS levels. So basically, my thinking is like they should be able to detect in the plasma, because in order for these enzymes to be functional, you also need to penetrate other organs, not just the liver. So they were saying if it's not in the blood, if it's just in the liver, it's not good enough. And Sandy said, yep, you're right. He's the president and CEO. The analyst from Barclays was like, if the liver, right, yeah. So I think in a way we do need to see some meaningful levels of plasma enzyme activity. And the president and CEO said, see, you're absolutely right. For the gags to be reduced, there has to be some production, I guess, into the plasma against other tissues. And that's why we're going to create a more sensitive assay, and that's going to give us all more comfort, because we'll see it in transit from the liver to the other tissues. Blah, blah, blah. This is actually somewhat important. Someone asked me yesterday, Gina, would I rather have seen enzyme activity or a new effect on gag? I tried to explain away because the patients were already taking ERT, and an effect on gag with enzyme level activity that's low source, it's undetectable. And I think we both say we'd rather have the gag, because the gag reduction can only occur if the enzyme is there. and the enzyme can only occur if the editing occurred. Do you agree? Yeah. We look at, so these are some calculations, right? So he mentioned how they're working on making a more sensitive test, essentially, more sensitive assay. And the test they use can only detect 5.2 nanograms per milliliter of enzyme in the blood. Now, these are some calculations that someone else made, but I went through them and it makes sense. So essentially, it's a bunch of math. It's already 30 minutes long, so I'm not going to get into it too much, but they basically used other gene editing data from the same company, same technique, and they did some math where you figure out what was able to be produced by a certain percentage of edited cells in monkeys, and you have to correct for things like the molecular weight of the two different molecules that are being made and whatnot. But essentially, at first it seemed like cohort 2 should have plenty, 15 versus detectable 5.2. But the issue is, I believe the calculation methods above are reasonable, but I did find a problem with starting assumption that this is like a number saying the levels in the dose will produce 33% of normal in monkeys. Unfortunately, the 33% of normal estimate initial use may have come from a higher dose and not the 5e13 animals. So essentially, to get these calculations, these would be more reasonable for like a higher dose. So if we actually look at the data and do different calculations with the actual right dose, comparatively. So, if we were to do the math, to sum it up, if we were able to do the math to figure out what amount of enzyme should be in the blood given the dosage given to cohort 2, we're looking at 2.3 to 4.6 nanograms per milliliter. Now, keep in mind that you look at this number and the assay or the test is only detectable to 5.2. So the theoretical amount of enzyme in the blood, even on the high end, is still lower than the amount detectable, which means that in my estimation of it, and I have more reasons for this, the enzyme was produced. It was produced in low quantities, but those low quantities still reduced GAGs. So the reason why you didn't see any IDS enzyme in the blood was not because the liver cells weren't edited and it, didn't make it. It's because it was just literally too low for the assay to detect. And if you do the pharmacodynamics and model it, with math and stuff like that using similar data, you get a pretty low dose. And this is actually what management and doctors like Munzer, who are in the study, sort of guided to, which is measurable levels but less than 100% of normal. So, they literally said, they're going to be low levels, less than normal, but they still should provide some benefits. So ultimately, if these calculations are right, lack of detection would have been expected given the assay shortcomings. However, at 5 times dose, cohort 3 should produce easily detectable levels of IDS. So this is also from the 9.5 transcript. Got it. And this is an analyst, and I guess it is a matter of coming up with a better assay, or do you think that cohort 3 with long-term follow-up you'd be able to detect at that point? And then the CEO said it's both of them. We need to remember this is cohort 2, and we stated it's a cohort, and we started a cohort that's 5 times the level, so one hopes the effect would be replicated or greater within cohort 3. And then this is where he says the exact amount. Our assay is validated down to 5.2, and even at levels such as the one you have in patients, we would hope that the benefits to patients reflected in GAG would be able to demonstrate a change in IDS that would help buttress the story. So basically the Sangamo management was too hopeful that if they had GAG reduction, that would obviously be enough to prove that IDS was created, but the investors were too nervous and didn't really probably do the math out to figure out what to expect, and if it's reasonable that if gag reductions are seen, it's probably from edited cells. So finally, I'm going to skim through the rest of this real quick because this is the main point, and I don't want the video to go on much longer because I'm getting tired. In healthy patients, the uptake of the enzyme from the plasma is a slow receptor-mediated process. And furthermore, any IDS taken up by the process in healthy patients is enzyme that's leaked out. Therefore, in healthy patients, there's always some extra IDS in the plasma that's leaked out. It's always there and always gets taken back up. So basically, naturally, your body always has a very low level of IDS in the bloodstream. MPS patients receive a very large dose, but IDS from ERT does not hang around the plasma long enough to get taken up by the slow process. Instead, a lot of the enzyme is just sucked up by the liver, which has a bunch of what are called non-specific receptors. So the liver, obviously in the body, is a filter, which is why, like, when you go out drinking and you have a hangover the next day and people say it's bad for your liver, it's because the liver's filtering out your body. And your liver also, it does that by having a bunch of non-specific receptors that just sort of suck up everything. So these people are on enzyme replacement therapy where their liver sucks up a bunch of the IDS. So Sandy said, and a bunch of other key opinion leaders agree that the liver is a very satisfied organ thanks to the ERT. So if the liver is, completely satisfied or has already insanely low gag levels from the therapy that's currently being on, so if the enzyme by the edited liver cells was truly staying in the liver, like the shorts in call selling institutions and Twitter parody scientists would like us to believe, it's fun. I didn't write this part, but that's funny. There would be almost no urinary gag reductions at all in the second cohort because liver gags are already so reduced by ERT. Think about it another way around. Ask yourself if liver gags can actually be reduced by so much more of an amount caused. Basically, if liver gags can be actually reduced by 50 to 60%, why wouldn't these gag reductions have already happened from the ERT, which almost all goes to the liver? Wouldn't patients on ERT already have urinary gag levels 50 to 60% lower? Since, you can't say it's the liver that's responsible for the gag reductions and the enzyme isn't being spread throughout the body when the liver is the one that's getting all the treatment right now, which is why we're trying to do a different one. MPS patients would already even have lower urinary gag levels than seen at actual current baseline. It is impossible, essentially, to get these kind of gag reductions just by further treating the liver because so much IDS from ERT already gets sucked up by the liver. Blah, 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 blah. Also the fact that all the, cells in the body are sort of starving for it. It sort of also probably reduces the detectability in blood, since when it's in the blood, all the cells are pretty much, dying for this enzyme, and they have a bunch of receptors that are unused, that have been unused for a while. Sort of like if you don't have a tolerance to something, it's because there's a bunch of receptors on it, and it's a similar deal with the enzyme, probably, where because the enzyme isn't naturally made in the body, the cells are all starving for it. They have a bunch of receptors for it, which might pull the enzyme away from the bloodstream. Therefore, you're not going to see it in the bloodstream as easily. Never mind the fact that the calculated amount is below the detectable level to begin with. Another thing is that determined sulfate, or dimatinine sulfate, comes primarily, this is a slide right here, these are the different types of gags. If you look at the localization of DS, it's in the skin, blood vessels, and heart valves. So there was a huge reduction in DS, so it can't be just from the liver, which is basically the big short hypothesis that the enzyme isn't escaping the liver, or it wasn't being made and it was just a fluke reduction. DS gags were reduced by 32% in cohort 2. They're formed in skin, blood vessels, heart valves, and not the liver. Clearly, the in vivo protein replacement platform is working, and IDS must be making its way out of the liver and to other tissues to cause this. So, as far as how this impacts stock price, just the run-up in anticipation to it caused it to go from 15 to 19 to 25%, upswing. And previously, just off news that they were working with Gilead and Kite on a cancer thing, had the stock at 26, which is a 62% increase from, if it got to the previous high it was in March, it'd be a 62% upside right now. And that's even before any results came out. So if as we talked about before, once the platform is proven to work for one disease, it's insanely likely it's going to work for a bunch of other diseases. So, the MPS market might not be huge, but basically curing every disease and having a gene editing platform that works and has FDA approval instantly makes this company a multi-billion-dollar company. I also last year had pitched Avexis to everyone, which is a gene editing company, or they don't do gene editing, they do gene therapy, which is similar but not as cool because it's not permanent. Essentially they have one thing that they can cure, which is SMA, spinal muscular atrophy, and at the time Avexis was valued at 4.5 billion. I did a valuation, and basically I put them at between 12 and 10 billion dollars, and this last was edited on March 7th, and I think it was in April or something like that, they were bought out for $8.5 billion, so there was 100% upside there. So if a company that can cure one disease is worth 8.5 billion, what is a company that has a technique to cure tens and tens of deadly diseases worth? If you just say 10 and you scale it up, it should be about $80 billion. And Sangamo is a $1.63 billion company. So 80 divided by 1.63 gives you 49. So the stock price should be 49 times higher than it is, which puts it at $785 per share. So that's why I have a bunch of options that are long term to have some extra leverage on this. You look at CRISPR companies let's see here, Edit Stock, and this company they're in the discovery phase, and they have a similar market cap. You look at CRISP, which recently has been getting beat up, but it has a $2.14 billion market cap, and not that long ago, it was almost a $3 billion market cap, almost triple us, almost double us, or double Sangamo rather, and it's just sort of insane. Let's see, we're at 43 minutes. Basically, real quick, Jeffries put out a statement after saying street expectations were misaligned. Sangamo killed it on the measure of gag reduction. Blah, blah, blah. We believe IDS as a result is a necessary component of gag reduction. We wonder if SPF-derived IDS remaining intracellular is the reason. So they're saying, is it staying in the liver cells? I think I explained why it's not going to be. And they said that they might have a better way of testing in 3 months. You look at Agorafin, they say a pretty similar thing. Basically all positive. Not going to go through that too much. Barclays put out a statement, and they had a call that was sort of private. You had to call in and know a code. And they put out a statement saying, we see today's data as directionally positive and expect further dose escalation should increase enzyme activity to improve clinical benefit. Jeffries put out an update today or yesterday saying first that Sangamo is the most advanced gene editing company. We have spoken to we and those we have spoken with have not proposed a good reason as to how else the gene editing drug would produce the gag reductions in cohort 2 versus cohort 1. And, they talk about what's, coming up in the pipeline stuff. They have almost a $30 price target. I think that's conservative, but finally I'm going to take like 2 minutes to talk about basically Sangamo also could have a cure for cancer. I know it sounds ridiculous, but just watch this quick video.
The immune system defends the body against disease. T cells are one of its key soldiers, targeting infected or abnormal cells. But cancer can block those defenses. Now scientists are genetically engineering patients' own T cells to make them smarter and tougher at seeking out and destroying cancer. One version is called CAR T-cell therapy. T cells customized to zero in on a patient's specific kind of cancer. It all starts when a patient's blood is filtered through a machine that separates T cells and other white blood cells from the rest. Scientists mix the collected T cells with a virus that has been disabled so it won't cause illness. Instead, that virus carries genetic instructions for the T cells to grow an artificial receptor, called a chimeric antigen receptor, or CAR, that will track its cancer target and rev up for an attack. Millions of copies of those engineered cells are grown in a laboratory and given back to the patient intravenously. The first CAR T cells are being tested against types of leukemia and lymphoma that bear a marker, or antigen, named CD19. Once in the bloodstream, those CAR T cells home in on that antigen and grab hold. They release toxic chemicals that trigger a cancer cell's death. As one cancer cell dies, CAR T cells move on to the next, multiplying in the patient's blood until they're no longer needed.
So basically, with zinc finger, you're able to edit those T cells more effectively. They recently bought out a company that has very vast experience in CAR T cells, and they have a partnership with Gilead that I actually talked about in my presentation. Gilead bought Kite, which is a CAR T cell company, and then Kite received exclusive license to leverage Sangamo's gene editing technology, and Sangamo will receive up to $3.01 billion in payments, and then they're also going to get royalties on sales of a cancer cure. So literally all they have to do is make this happen, not even sell any cancer curing drugs, and they'll get $3.1 billion, which is double the market cap of the company right now. So they're going to get more than twice. Just having that would make the stock be twice as high as it is now. And then obviously if you get royalties on a cure for cancer, I'm sure that's not going to bring in no money. Yeah, so that's pretty much it. Here's a paper about Sangamo and CAR T cells and cancer. I'm not going to get into it too much, but essentially results suggest that this gene editing approach can support a more potent anti-tumor effect while potentially eliminating the risk of essentially they're able to prevent allergic reactions and so on and so forth. But yeah, cure for cancer, cure for a bunch of different diseases. No safety issues so far, unlike CRISPR, which is actually causing cancer. CRISPR causes cancer. You can Google that and read the studies about that. So yeah, I know this is going to be a 50-minute long video, but it's sort of a complex topic, and I'm going to be joining the RAM Fund again in spring. I have a bunch of computer science classes I'm taking now, so I don't want you guys to sell my position while I'm not there, and hopefully even add to it like I am. I'm not going to show you how much money I have in my account, but after sort of being blown up on the 5th by being over leveraged in options, working overtime and buying more for January and February, since it's going to be good news with the 5 times higher dose, and they could announce that they've detected enzyme any day really, since they're working on developing a more sensitive test in the first place. And never mind, I've not even talked about whole other areas that they're doing, like gene therapy for hemophilia A, where it's not gene editing. They have a gene therapy solution that's better than all the other competitors, but like Biomarin and Spark are having issues with theirs. Sangamo's not. That's like a $25 billion industry that they could have at least a third of per year, so on and so forth. But I think that's it. If you guys have questions and you're in RAM Fund or something, have Professor Ice give you my email. If Professor Ice, if you have questions, email me. A lot of the information I showed was mine. A lot of it was not mine, and from various sources online and discussion groups. that I frequent and whatnot, but hopefully I sort of gave an understanding of it. All right, yeah, I'll hopefully you guys buy more, and if you're not in a RAM fund and see this or something, maybe you should buy Sangamo, whatever. Disclosure, I am invested in it, whatever. All right, see you later.